Genomic stability is a fundamental requirement for cellular survival and organismal integrity, maintained through a coordinated network known as the DNA damage response, or DDR. This system detects genomic insults such as double-strand DNA breaks and replication stress, and initiates signaling cascades that determine whether a cell repairs the damage or undergoes programmed cell death. At the core of this process are the sensor kinases ATM, which stands for ataxia-telangiectasia mutated, and ATR, which stands for ataxia-telangiectasia and RAD3 related. ATM is primarily activated in response to double-strand DNA breaks, while ATR responds to replication stress and stalled replication forks. Upon activation, these kinases initiate downstream signaling through checkpoint kinase activation, specifically CHK1, checkpoint kinase 1, and CHK2, checkpoint kinase 2. These checkpoint kinases propagate the DNA damage signal through phosphorylation-dependent mechanisms, ensuring that the cell does not proceed through the cell cycle with damaged DNA. This checkpoint kinase activation cascade functions as a critical bridge between damage detection and transcriptional response. One of the primary targets of this signaling network is the tumor suppressor protein p53, also known as tumor protein 53. p53 becomes stabilized through post-translational modifications, including phosphorylation at serine 1,5, which prevents its degradation. Stabilized p53 accumulates in the nucleus and functions as a transcription factor, activating genes involved in cell cycle regulation, DNA repair, and apoptosis. A key downstream effect of p53 activation is the induction of p21, also known as cyclin-dependent kinase inhibitor, 1A. p21 inhibits cyclin-dependent kinases and enforces cell cycle arrest at the G1 to S phase transition. This pause allows the cell to engage DNA repair pathways and restore genomic integrity. If repair is successful, the cell resumes normal function, and genomic stability is preserved. If the damage is extensive or irreparable, p53 dependent pathways trigger apoptosis, a programmed cell death mechanism that eliminates damaged cells. Failure of this system leads to mutation accumulation, chromosomal instability, and increased risk of oncogenic transformation, which refers to the process by which normal cells become cancerous. These outcomes are central to cancer development and progression. This pathway does not operate in isolation. It is influenced by crosstalk with other biological systems, including redox signaling and epigenetic regulation. Reactive oxygen species, or ROS, can induce DNA damage, while epigenetic modifications, such as DNA methylation and histone modification, can alter the expression of DNA repair genes, further compromising genomic stability. Within a systems biology framework, genomic stability interacts with inflammatory signaling, metabolic regulation, and cellular aging processes. Understanding this integrated architecture provides a foundation for interpreting disease mechanisms and identifying therapeutic targets. Extended mechanistic mapping, annotated study synthesis, and translational analysis are available in the accompanying scientific protocol. The expanded material includes detailed modeling of ATM and ATR activation dynamics, checkpoint kinase signaling networks, p53 transcriptional regulation, DNA repair pathway integration, and cross-domain interactions involving redox signaling and epigenetic modulation, alongside structured summaries of molecular and translational research. This material is provided for educational and scientific purposes only and does not constitute medical advice. Clinical decisions should be made in consultation with qualified healthcare professionals.